My name is Dr. Pradnya Kedari. In this module, we are going to study about transport across the membrane. A cell constantly needs to interact with its surrounding in order to maintain the balance inside the cell. To maintain this homeostasis, cell need to control over what goes outside and what comes inside the cell. One very important structure called cell wall maintains this transport. In this module, we are going to study regarding detailed structure of cell wall and its types of transports. Learning objectives of this modules are introduction, cell membrane and cell wall, function of a cell wall, basic structure of cell membranes, transport across cell membrane and its types, active transport and passive transport. Some important terms to remember. Homeostasis, it means internal balance. Transport, it means movement of materials. Solute, the substance being dissolved or a dissolved substance in a solution. For example, sugar, salt. Solvent, the substance which is dissolving the solute. For example, water. Solution, a mixture in which two or more substances are mixed evenly. Concentration, the amount of solute in a solution. Concentration gradient, the gradual difference in the concentration of solutes in a solution between two regions. Food and water move into a cell. Waste moves out of a cell. Cells need to constantly move the molecules across the membrane and try to reach equilibrium, that is, equal balance. During transport of material across membrane, free energy of solute depends on the concentration gradient across the membrane. In case of uncharged solutes, solutes move from higher concentration to compartment of lower concentration spontaneously. Presence of a membrane potential as well as the chemical concentration gradient play very important roles in distribution of ions. Cells need to move molecule constantly across the membrane in order to try to reach the equilibrium. Actually, such equilibrium is never truly reached. Cell membrane and cell wall. All cells have a cell membrane made up of protein and lipids. Some cells have cell membrane and cell wall. For example, plants, fungi and bacteria. Plant cells have a cell made up of cellulose. That cellulose is fiber in our diet. Bacteria and fungi also have cell walls, but they do not contain cellulose. Cell membranes and cell walls are porous, allowing water, carbon dioxide, oxygen and nutrients to pass through easily. Why cell membrane is necessary? Cell membrane is necessary because it is one of the multitasker. It delimits the cells, it provides structure for a cell, it defines internal components of eukaryotic cell, and cell membrane protects cytosolic content from the environment by separating the interior of cells from its environment. It acts as a barrier for freely diffusible water-soluble molecules present in the environment. So, no particle can randomly enter the cell. In short, it controls the entry of any molecules into a cell. It maintains the delicate homeostasis of each cell. That is why cell membrane is very important. Function of a cell membrane. Cell membrane separates the component of a cell from its environment. They are the gatekeepers of cell. They regulate the flow of material in and out of the cell. Cell membranes are selectively permeable. Cell membranes help a cell to maintain homeostasis, that is, its internal balance. But how cell membrane does it? Well, All the cell membranes consist of bilayer of polar lipid molecules and associated proteins. Protein allow the selective transport of various molecules. They transmit signal across the membrane and much more. These membrane proteins have many specialized functions to perform. For example, they act as receptors. They are involved in the selective transport across the membrane. They participate in electron transport, oxidative phosphorylation, cell-cell interaction in multicellular organism, etc. To understand how this actually takes place, we need to know the structure and components of cell membrane. Let's see basic structure of cell membrane. The fluid mosaic model describes the molecular organization of cellular membranes. This model suggests that the structural organization is made up of 1. Membrane lipids, that is phospholipid bilayer and associated proteins. Let's see details of membrane lipids. Lipid make up most of the part of cell membrane, approximately 10 raised to 9 lipid molecules per cell membrane. These are made up of phospholipids, glycocerebrosides, galactosylglycerides and sterols. These molecules are amphipathic in nature. It means they have both hydrophilic and hydrophobic regions. Membrane lipids are made up of 2 hydrophobic fatty acid chains or tails, which are linked to a phosphate containing hydrophilic head group. The fatty acid tails are poorly soluble in water and therefore are called as hydrophobic. The head groups are polar and are exposed to water on both the sides. Therefore, they are called as hydrophilic. When membrane is brought in contact with water, the hydrophilic head group maximize their interaction with water molecules and hydrophobic tails try to minimize the contact with water by interacting with each other. The geometry of resulting lipid assembly balances between hydrophilic and hydrophobic domains. This bilayer represents the basic structure of all the biological membranes. 50% of the mass of most of the cell membrane is lipid, but this content can vary depending on the type of organelles or cells they are present on. For example, mitochondrial inner membranes have many protein complexes needed for electron transport and oxidative phosphorylation, and therefore 75% of this membrane is made up of proteins. Plasma membrane of animal cells contain additional glycolipids and cholesterol, which corresponds to about 40% of the total lipid molecules. Phospholipids. They are associated with inorganic phosphates. Phospholipids are abundantly present. It has polar head and 2 hydrophobic hydrocarbon tails. One arm has 1 or more CH double bond CH, due to which there is kink in a tail. Mobility of phospholipids in a membrane. As lipid molecule in a lipid bilayer are not bound to each other covalently, they can move freely. Due to this, the membrane can be flexible to assume any shape without disrupting its hydrophobic interactions. Phospholipids can bob up and down, rotate, diffuse laterally and flip and flop too. A flip is a rare event as it needs a polar head to migrate through hydrophobic interior part of the bilayer. Some membrane contain an enzyme flippase which catalyzes this movement across the bilayer. Different flippase enzymes are required to flip lipid substances in a particular direction. Individual molecules of lipid bilayer, that is, lipids and proteins, have ability to rotate and move in a lateral direction within the bilayer. It is possible and dependent on temperature and lipid composition. For example, the shorter fatty acid chains have weaker interactions as compared to that of the longer fatty acid chains. Therefore, membrane containing shorter fatty acid chains are less rigid and can remain in fluid state at lower temperatures also. In case of unsaturated fatty acids, double bonds introduces twist in fatty acid chains, making it difficult to pack them in a bilayer. Glycolipids. These show presence of carbohydrate groups. These are present in small amount and usually on outer side. Phosphatidylcholine. These are the lipids with presence of choline in head group. These are present only in the outer half of membranes. Liquid with terminal amino group like phosphatidylethanamine and phosphatidylserine are present. This gives negative charge to the inner side of the membrane. Membrane proteins. Biological membranes consist of proteins inserted into the lipid bilayer. Protein is a major constituent of cell membrane other than lipids. Amount of proteins are variable. It constitute about 25 to 75% mass of various membranes of a cell. The current model of membrane structure was proposed by Jonathan Singer and Garth Nicholson in 1972 and is called as fluid mosaic model of membrane. According to this model, proteins are inserted into the sea of lipid bilayer. Phospholipids form basic structural organization in this model. Membrane proteins from this structure carry out the specific functions. There are types of proteins associated with membranes and are divided into two general classes as integral membrane proteins and peripheral membrane proteins. Integral membrane proteins are also called as transmembrane proteins. They are directly inserted within the lipid bilayer and part of these proteins are extended out on both the sides of the membrane. The hydrophobic side chain of these amino acids interact with the fatty acids chain of membrane lipids. Transmembrane proteins are amphipathic molecules. That is, they have both hydrophobic and hydrophilic regions. Hydrophilic portion of these proteins are exposed to aqueous environment on both the sides of the membrane. Some transmembrane proteins span the membrane only once, which are known as single pass, while others have multiple membrane spanning regions known as multipass. Most transmembrane proteins of eukaryotic plasma membranes are modified by the addition of carbohydrates. These proteins are exposed on the surface of cells and they may participate in cell-cell interaction. Another class of protein is peripheral membrane proteins. They are associated with membrane indirectly and are not inserted into the lipid bilayer. These proteins generally show interaction with integral membrane proteins. Some proteins are anchored on the membrane by covalently attached lipids. Such proteins are anchored to extracellular phase of plasma membrane by glycolipids and to cytosolic phase by fatty acids or prenyl groups. Probable function of these membrane is intracellular signaling. Some proteins are exposed to external surface attached to lipid bilayer through covalent linkage via oligosaccharide like inositol or mannose. This was all about the structure of plasma membrane. Now let's proceed to transport across the cell membrane. Transport across the cell membrane. Due to selective permeability, a cell can control and maintain the internal composition. In a cell, small uncharged molecule can diffuse freely through phospholipid bilayers. Small non-polar molecules, for example O2 and CO2, are soluble in lipid bilayer, so can cross over cell membranes easily. Small uncharged polar molecules, for example H2O, can diffuse through membranes easily. Larger uncharged polar molecules, for example glucose, cannot diffuse through membrane. Charged molecules, for example ions like hydrogen ions, hydrophilic solutions like sucrose and amino acids cannot diffuse through a phospholipid bilayer regardless of its size. The molecule which cannot diffuse across a lipid bilayer, for example glucose, can also enter through the membrane by the action of specific transmembrane proteins. These transmembrane proteins act as transporters. Therefore, they play critical role in the membrane function. They show multiple spanning region in a molecule which forms a passage through the lipid bilayer. Polar or charged molecules can cross the bilayer through this passage without interacting with the hydrophobic fatty acid chains. These membrane transport proteins belong to general classes as follows: ATPase or ATP powered proteins, channel proteins and co-transporters. Some of these proteins are very well characterized, for example aquaporins. On the basis of requirement of energy for transport, there are two types of transports across the membrane. First is active transport, which requires energy. It can occur against concentration gradient. Energy is required as molecule must be pumped against the concentration gradient. Proteins that work as pump are called as protein pumps. They can move large molecules and charged molecules, for example Na plus, K plus, Cl minus, etc. Example: body cells must pump carbon dioxide out into the surrounding blood vessels to be carried to the lungs for exhalation. Blood vessels are high in carbon dioxide as compared to the cells. So, energy is required to move the carbon dioxide across the cell membranes from low to high concentration. Types of active transport in a cell. They are as follows. 1. Pump. Sodium potassium ion pump. This is where the concentration is moving low to high concentration. Uses protein in membrane to pump ions against concentration gradient. Ions are elements with a charge plus or minus. Carrier proteins. They bind to specific molecules and move them from area of low concentration to an area of high concentration. Channel proteins. They form open pores through membrane. The molecules of appropriate size, for example ions, can cross the membrane through these pores, for example ion channels. Channel proteins form a pore through which ions of appropriate size and charge can pass the membrane by free diffusion. Channels are opened and closed according to extracellular signals and keep control on the movement of ion transport across the membrane. It allows inorganic ions like Na plus, K plus, Ca2 plus and Cl minus to pass through the membrane. Carrier proteins. They act like enzymes. selectively bind small molecules to be transported. They undergo a conformational change. Change in the conformation opens a channel. The molecules to be transported can pass through these channels and are then released by carrier proteins on the other side of a cell membrane. This transport can be associated with energy changes coupled for production of other forms of metabolic energy. What is the difference between channels and carriers? Channels, they open on both the environment of the cell, that is extracellular and intracellular at a time. It allows entry of millions of ions per second, facilitate diffusion without any interruption. Channels and pores do not have any binding sites, whereas in case of carriers, they open up only on one side, that is either extracellular or intracellular at a time. Only 100 or 1000 ions can pass through per second. Carriers have binding sites. Carrier proteins are very specific and can bind or transport only one or a group of very similar molecules. Defects in carrier protein can be correlated with specific disease. Carrier proteins are of various classes. Endocytosis and exocytosis is the mechanism by which very large molecules such as food and waste get into and out of the cell. Endocytosis. It's a movement of large molecule into the cell. During endocytosis, membrane starts to make an inward fold. Large molecules move into that opening or pocket and membrane surrounds the molecule with bilayer, creating a vesicle. Vesicle, that is molecule inside it, moves into the cell. Membrane gets sealed up. White blood cells, which are part of immune systems, surround and engulf the bacteria by endocytosis. White blood cells, which are part of immune system, surround and engulf the bacteria by endocytosis. Endocytosis. Large particles are engulfed by a portion of cell membrane and the membrane then breaks off and travels into the cell, carrying the large particle with it. It is of two types: phagocytosis, where food material is taken inside, and pinocytosis, where water material is taken inside. Exocytosis. Movement of large molecules out of the cell is known as exocytosis. Molecule surrounded by bilayer vesicle, that is from Golgi, membrane absorbs bilayer vesicle, and third, large molecules are pushed out of the cell. Passive transport. Passive transport does not require any energy. It occurs according to concentration gradient simply by diffusion. It occurs by following ways. Diffusion. Diffusion is movement of molecule across the membrane down the concentration gradient. High to low concentration, spreading out the molecules. Always trying to reach equilibrium, that is molecules are evenly spread out. The more kinetic energy, the faster is the rate of diffusion. More is the temperature, faster is the rate of diffusion. Facilitated diffusion. Diffusion of a substance across the membrane with the help of protein embedded in the membrane is known as facilitated diffusion. Movement is assisted and still moving molecules from high to low concentration. Next is osmosis. The diffusion of water molecule across the membrane is known as osmosis. Movement of water from area of high concentration to area of low water concentration. It is always trying to reach equilibrium, that is molecules are evenly spread out. Movement of water from area of high concentration to area of low water concentration. always trying to reach equilibrium. Passive type of transport takes place in following molecules: small molecule, uncharged molecule and water. Gases like oxygen and carbon dioxide. These were the details regarding components of cell wall, its structure and different type of transports. In the next module, we will be studying in detail regarding different type of transports. Thank you.